Hello, and welcome back to the lecture on Chapter 7, Microbial Genetics. We had just finished up Part 3. We had talked about processing the messenger RNA in eukaryotic cells. So this generally does not happen in the prokaryotic cells. So the eukaryotic cell, the situation is a little more complex. There's this messenger RNA that gets edited before it can leave the nucleus and gets that guanine cap and poly A tail. Things are a little bit simpler in the prokaryotes. We get this messenger RNA. So we get transcription, get the messenger RNA, and then that's it. As soon as a ribosome can connect to this, translation can happen, and we get a polypeptide. Remember, that's a bunch of amino acids hooked together in a row. Now we're going to see how it happens. It's the process by which these organelles called ribosomes, largely made out of ribosomal RNA, they take that information that had been coded in the DNA, and now we're going to use that information to figure out which amino acids to put in what order to make a polypeptide. This is that famous table of all the 64 codons. So these would be sequences on the DNA, and then, or sorry, not on the DNA, on the RNA, and then what amino acid is associated with that. You've seen this table before in plenty of biology classes. The thing is, this is a universal table. All of the species that have DNA as their genetic information use exactly the same table. The ribosomes and the transfer RNAs working together use this same table. I'm going to show you a little video about these codes. Oops.
The 4 nucleotides of messenger RNA are arranged in 64 three-letter words called codons. The genetic code is a language consisting of these 64 codons. 61 of the 64 codons code for a specific amino acid, while the remaining 3 instruct the ribosome to stop the translation process. Let's see how this table is used to determine the amino acid of a given codon, CAG. First, on the left side of the table, find the first letter of the codon, C. Then on the top of the table, find the second letter of the codon, A. Finally, locate the codon that has a G as its last letter, and you will see that the amino acid coded for by CAG is glutamine. Can you determine which amino acid is coded for by UCA?
So UCA, you're going to look for the first codon over here, U, second codon, C, over here, and then finally, third codon, A. There we go.
Serine is correct. Now, can you determine the effect of UAA?
So we had UCA, and now if we change that middle letter to an A, that's going to be a really
The codon UAA signals
mutation.
termination of the elongation of the polypeptide. It's a stop codon.
You may remember from your biology classes when we have a mutation like that that doesn't lead to a different amino acid, but instead a stop codon. Those are the nonsense mutations. Missense would be if it brought a different amino acid. So you just remember mutations happen, and this is the mechanism that mutations have these different effects depending on what is coded for. Okay. So the participants in this process of translation, you've got the messenger RNA that has the genetic information that it like picked up as it was being transcribed from the DNA. We need 2 more really important players, and those are the transfer RNA molecules. Those are going to bring the amino acids to the messenger RNA. And finally, the platform on which all of this is happening is ribosomes, which are made out of ribosomal RNA. Transfer RNAs first. It's just a strip of RNA. So like long thing that then folds onto itself. We represent it in a number of different ways, kind of like this clover leaf shape. Once the thing is folded and set up right, there's that little section down here. Now, all of these are letters, but it's this little section down here that's got to match up to the codon. And it's been given this unfortunate name of anticodon. It just means this is the bit that's going to be complementary to the codon, and that's how it's going to bind to the messenger RNA. And then at the other end, it carries the amino acid. So one of these charge transfer RNAs has an amino acid here to bring into this process of translation. I'm going to sneeze, excuse me. Sorry, excuse me. All right, so ribosomal structures. Here's an important point to understand. There are a couple of ribosomal subunits that have to come together. I think of them as hamburger buns, have to come together. There's a small subunit and a large subunit. And they're a little bit different in prokaryotes compared to eukaryotes. And so these, I think I may have mentioned this previously, these make an excellent target for medications because we can impact the bacterial translation and their ability to make the proteins that they need without impacting our translation, the eukaryotic cells. And that's why understanding this level of detail helps us to understand how medications work, or in one day, perhaps, if that's something in your future, that you can start developing medications that help fight off infection without hurting the human cells. And assembled, put all together, we have the messenger RNA, that's the strip. Then we have to have the two subunits, the small ribosomal subunit and the large one. And then we'll see in a video, there's all these different sites and they have letters and super confusing. Here's the easy way to remember it. A stands for the amino acid site. So let's go back to this for a second. It's where one of these transfer RNAs is going to arrive holding on to its one amino acid. Then remember, we said when you put a bunch of amino acids together, that's called a polypeptide. So once this amino acid gets bound to the next one over, we end up with a chain of amino acids together. The transfer RNA that's still holding on to that chain, that's in the polypeptide site. And then finally, when that transfer RNA is ready to leave because it has left, it has released its own amino acid, then it can exit through the E site, the exit site. Yeah. So arrive and exit, or amino acid exit, polypeptide at one in the middle. Okay. We'll see that in a video in the next section. See you over there.